Hello, everyone. My name is Chang Lu, a postdoc from Science Lab. It's a pity that I'm not able to attend on-site due to a visa delay, but I'm excited to share our latest research with you virtually, which is about the integration of cellular circuits and immune repertoire at spatial level. T and B cells play a key role in the adaptive immune system by recognizing the antigens through PCR and TCR. They are diverse in terms of functions and their interactions with partner cells. For example, this picture shows the activation of B cells by T helper cells. So understanding how T cells and B cells function and communicate with other cells is crucial for treating various diseases, including autoimmune disorder and cancers. However, most of studies are still limited at a gene and a single cell level. Therefore, in this work, we aim to design a strategy to explore the functions of T and B cells, as well as their interactions with other cell types at the spatial and the clonal level. So, as we already know, BCR include light chain and heavy chain, and TCR is straight with alpha and beta chain. They are hypervariable due to a procedure called the VDJ recombination. The random joint of VD and J gene segments with a highly variable CDR3, contributing significantly to the diversity of antigen binding sites. As most of studies currently for this work, we only tried the BCR heavy chain and the TCR beta chain because they tend to be more diverse than light chain and the alpha chain due to the inclusion of the D segment. The diversity of BCR and TCR can be measured with VDJ sequencing. The pipeline is what bioinformaticians usually do. They translate the nucleotide sequencing into and then compare the similarity between the sequences or embedded the sequence into a vector with the same length to identify the relevant clonotypes or query or predict the relevant epitopes or antigens. Some recent developed single-cell sequencing profiling enables the generation of paired transcriptomics and immune repertoire data at a cellular resolution. Then some studies attempt to integrate paired transcriptomics and VDJ information, understand the interdependence between these modalities by projecting the cells from different modalities into a joint latent space. However, a technical limitation here is obviously it contains no spatial information. And we all know spatial info can provide valuable insights into the microenvironments and the cellular heterogeneity. To solve this problem, last year, I had the honor of being involved in the Spatial VDJ project, which was led by Karolinska Institute and KTH in Sweden. Spatial VDJ is an extension of Wisdom spatial transcriptomics that can map gene expression as well as VDJ sequences within tissue. However, there is still a lack of effective methods to study how T and B cell functions and interact through this spatial multimodal data. So this is our motivation and objective. A straightforward idea to do this is we first integrate spatial VDJ with spatial transcriptomics and then calculate the association between variables. So we consider if the single cell VDJ integration method that we mentioned in the previous slides could be used to solve this problem. Unfortunately, they are not adaptable for two reasons. First of all, these methods aim to clustering or classify cells, but our task is to associate variables. Secondly, interacting cells are usually close to each other, but this method did not consider this part. We then thought if we could use multi-omics integration method to answer this question. So we tried a factorization-based unsupervised framework called MOPA on the spatial VTC data from 6 slides of human tonsils. These 6 slides were collected from the same person with different distance from each other. We input 3 counter matrices, IGH, TRB, and the gene expression into MOPA, and hope to infer an interpretable low-dimensional representation in terms of a few tenant factors. That would help us find some significant association between features. We clustered IGH and TRB clones based on their feature weight matrix, but unfortunately, we did not find any positive related clusters. The main reason is that the TCR and the BCR matrices are much sparser than the gene expression matrix. This results in a low spatial overlap between IGH and the TRB. In addition, this method completely ignores the clone sequencing information. It seems that the current approaches could not be used directly to solve my problem. Therefore, we proposed a new strategy based on multi-layer network community association for joint modeling of the immune repertoires and the transcriptomics as the spatial resolution to understand the role of immune repertoire in intra and intercellular signaling. Sorry, I think there's something wrong. I will start from here.
We are seeing it fine. If you go back to yes.
Yep.
Yeah. Then let's dive into the framework. For individual human tonsil sections, we have not only spatial transcriptomics and spatial VDJ count matrix, but also the VDJ sequencing data. Since spatial VDJ is based on visual, which means a spot may include multiple clones. Based on this data, we aim to design a method to understand the interaction and the signaling between B and T cell subsets. For spatial transcriptomic data, we perform the cell deconvolution to obtain the proportions of cell types on each spot and generated cell type network. And for spatial VDJ data, we construct IGH and the TRB clone network based on their sequence similarity and spatial localization. Next, we link these 3 networks into a multi-layer network, where the similarity across network are computed based on their localization of the tissues. Eventually, the cell interactions can be inferred by calculating the association between the communities or say clusters across layers. The framework can be divided into 2 steps: network construction and multi-layer network community association. And there are 2 things I need to keep in mind. As I mentioned before, I need to consider both sequence and the spatial similarity and to address the problem of sparsity of spatial VDJ data. To preserve the spatial and the sequencing information for TRB and IGH data, we first constructed a graph based on spatial count matrix using the mutual nearest neighbor, and then embedded the HH or TRB sequencing for each clone using ISMB. We then use a variational graph autoencoder to reconstruct the IgH and TRB graph. The idea behind it is if two clones were localized and their embedded sequencings are very similar, they will be linked on the graph. For the gene expression layer, we constructed a cell type network based on cell type proportion. We then can add cluster clones using graph-based community detection methods on individual graphs or cluster clones on the latent space from the autoencoder. Next, we calculate the Spearman correlations between the mean of the community cross layers. By merging the sparse data within communities and calculate the overall correlations between communities, the problem of clone count sparsities can be mitigated. Let's say if this idea work. We identified 28 IgH clusters and 21 TRB clusters from individual layer, and cluster the Spearman correlation between these clusters and the cell type at the spatial level. For the IgH heat map, we can see IgH clusters are divided into two distinct models. One is related to the cell types within germinal center zone, and the other is related to the cell types outside the germinal center zone. In addition, from both heat maps, we can find some IgH clones colocalized with some T cell subsets and TRB clones colocalized with some B cell subsets. This indicates that the interaction might happen between these B and T cells. However, with current pipeline, it's hard to find any significantly positive correlation between IgH and TRB clones clusters at the spatial level. This is because the clusters were defined on each layer independently. Therefore, we need a strategy which is able to detect the communities based on the connections within and across layers, and also calculate the association between the communities cross layers. We then need to go back and consider how to solve the problem of low overlap between the IgH and the TRB layers due to the sparsity of the count data. Here we use the Morans R correlations, which can be thought of as a weighted version of correlation, where W is a weight matrix based on the Euclidean distance on spatial coordinate. The idea behind Morans R is, instead of only considering the overlap of IgH and TRB clones, if an IgH clone and a TRB clone are next to each other and their counts are relevant, they may also have interaction. Since our data were from the same tonsil, the distance we used are 3D Euclidean distance. Thus, the link between two layers can be generated. Now we have already constructed a 3-layer network. Next, we implemented deep multi-graph clustering, which is proposed by Luo in 2020 to detect communities that's spatially colocalized. Here, I only show how the method detects communities between IgH and the TRB layers, but it can be extended to more layers, of course. We first map clones on IgH and the TRB layers to latent space via an autoencoder. The proximities between clones in individual graphs can be pursued by optimizing the intra-layer and intra-layer loss function. The IgH or TRB clusters and their centroid can be obtained via a clustering loss based on a minimal entropy. Meanwhile, we defined a cluster association matrix C from a unified space C to update the cluster centroid mu from the latent space of IGH and TRB using an attention-based function. The idea of this step is the more related the two clusters from different layers are, the more shared edges they have. So the updated mu is able to capture cross-graph dependencies at the cluster level. By minimizing the joint loss functions, we can get the cluster matrix Q for each cluster, for each graph, as well as the association matrix between graphs. Here are some results. We integrate IGH, TRB, and the cell types layers and performed the framework. As you can see from the heat map, we found the significant association between regulatory T cells and the TRB cluster 3. In addition, we found the TRB cluster 3 is associated with a group of IGH clusters from the heat map on the right side. By visualizing the clone clusters and the cell proportion on the HND images, we found that the regulatory T cells and the TRB cluster 3, they are overlap at many spots, especially on the bottom region. And in this region, many clones of TRB cluster 3 either localized or are close to the IGH clones we identified. We then queried the IEDB database using TCR match to find the epitope matches and specificities. Some antigens like ORF7b and ORF1ab were hit, but still need to be verified by immunologists. For the next step, it will be interesting to validate what these clones are. So we will do the downstream analysis, including epitope prediction, querying the TCR and BCR database, and the HLA matching between the predicted epitopes and the patients. In addition, we are applying the framework on another spatial VDJ data from breast cancer and the spatial TCR data from Benotman's protocols to see the applicability. Furthermore, we are using a cell deconvolution method called DOT, which is proposed by one of our lab members to do not only cell deconvolution, but also gene expression estimation for each cell type. So the next step is to generate and integrate cell type-specific co-expression network into this multi-layer framework to study the intracellular signaling. In the future, this framework will work with Liana, all-in-one cell communication tool from our lab. It integrated existing methods and knowledge to study single cell and spatial cell-cell communication. And also will work with decoupler, which is an example of methods to infer biological activities. In conclusion, understanding how BCR, TCR, and the gene expression interact is important to improve our knowledge of immune response, disease diagnosis, and therapy development. Current methods for integrating single-cell VDJ and transcriptomics are not well suited for spatial VDJ data. Our approach combines the spatially resolved VDJ and the gene expression data based on both sequence and spatial similarity to find the related cell types and clones. Currently, we are adding new functions to analyze intracellular signaling and antigen specificity based on public database. Thank my supervisor Julio, my collaborator Camila Kim, Qirong, and Jonah Kim for their efforts, as well as the help from our lab members. If you are interested in the Liana Plus and the spatial VDG, please check out the link here. And it is still an ongoing project, so I'm happy to receive any suggestion. Thank you.